Hi, everyone. I have had to abandon Wi-Fi. The only place where we can plug directly into the internet is in the music room of my home. So if you notice a lot of instruments behind me, there's a reason for that. I'm at Brandon's desk, and this is not the ideal system for lighting, but I have a ring light that I think you can still see models and stuff with. So I've tried to get that fixed. Okay. I know some of you might not be with me because I told you I was just going to try again. I sent an email in case you still wanted to try and tune in. I understand that that's not a possibility for everyone right now. So anyway, here we go. I'll have to try and work on whatever's going wrong with the Wi-Fi here. Okay, so I'll go ahead and just go ahead and get started, I guess. I wanted to I had to basically redo all my settings, so give me a second. Hi, Conrad. Please let me know if this is any better. I think it's probably better, but please give me some feedback if you can. I appreciate you guys being patient with me. This is really frustrating when you're all, I can't look at you and tell you what's going on. Okay, so where did I put my writing tablet? I basically ran upstairs and okay. Okay, good. Yeah, there's something wrong with my Wi-Fi. Okay, that's awesome. All right. So have to fix everything. Hi, Steven. I'm not talking to you. Sorry, he's still thinking I'm having problems. Hi, Steven. Yeah, so I have a meeting at 12:20 today. So I have to be on something else to have that meeting, but I'll be able to answer questions, or if you need to schedule a time with me, you know, to chat or whatever online, that's fine. Just message me and let me know. We can set that up. Let me make sure. Nope. All right. So hi, at least Stephen and Conrad. It looks like you two are both here. Again, sorry for the delay and the problems. I was going to actually start off the talk today talking again about sugars and glucose in particular, since that's the one we're going to spend so much time focusing on. And so I need to pull up my camera to make sure you guys can see what I'm doing. Okay. Do it this way. All right. So first off, when you think about glucose, sorry, this is completely inverted from how you guys see it. Like my camera flips things. So you usually see the glucose kind of drawn up here in the top, or the oxygen drawn in the top right-hand corner of whatever Haworth projection you're usually looking at for glucose. And we tend to draw it like it's flat, but I want you to remember back to organic chemistry how we actually form chair conformations. So if you can kind of look there and see the chair conformation. I didn't put all the hydrogens on here, just the OH groups, and then the CH2OH right here. But I just wanted to kind of remind you that even though most biochemists draw it in a Haworth projection, making it look like it's pretty flat, it actually looks like that, okay? And this flips back and forth between the two chair conformations where all the OH groups are in the axial position and then they flip to being all in the equatorial position. So I want you to remember that when they're in the equatorial position, that's where they're the most stable because it lessens the 1,3 diaxial interactions, okay? If you need me to go into more detail on those, I can make a quick video about that, but I wasn't going to focus on that for this video. Just kind of more like, remember stuff you learned in organic, it applies to this, but in a way that most biochemists never really look at. All right, so one of the things that I had you do after my lecture the other day was to think about the two forms of glucose. So I mentioned that there's an alpha form and a beta form. And these are when glucose is in the cyclic form, not when it's linear. So I've got up, down, up, down. And then if I have alpha glucose, it goes down. And then if I have, let's see, beta glucose, whoa, I can't see my cursor here at all. Oh. What's going on here? Oh, shoot, I see what I'm doing. I can't use two pens at the same time, I'm sorry. So if you have the OH pointing in the up position, then that's going to be considered beta glucose, all right? You can look up more information about these as far as stability and things like that if you want, but since we've already lost so much time this semester, I'm just going to kind of keep going on with it, okay? So alpha and beta glucose, and these are essentially in equilibrium with each other. Also going back and forth with the linear version. One thing that I do need you to know I need you to be able to draw both of these forms of glucose and know which one's alpha and which one's beta. And I need you to be able to draw the linear form that I put up on here the other day. Okay, so that's part of, oops, sorry. That's part of what I need you guys to be able to do. All right, so the next thing that I normally do when I start this conversation is to thinking about glycolysis. And one of the things that I usually like to do is ask the class as a whole, and obviously I get more answers depending on what specific classes the people in my class have had. So like, I know some of you have had cell biology already, some of you are in it right now. So if you've had cell biology, the animal physiology, if you've had a bunch of those types of classes, then you might remember or know more about glycolysis than the average student who hasn't had any of those classes. But usually what I do is I go up on the board and I go around the room and I just say, thinking about the word glycolysis, give me information that you know. Okay. And I usually can get about one board of information if the class is full of about, 25, 30 people. If it only has, 10 people like our class, I usually don't even get that much. I get like half a board of content because the truth is that the way that we tend to think about glycolysis in biology is much different than the way that we tend to think about it in chemistry. So, common pieces of information that if I had to guess, because I've done this before, if I asked you to come up with words, and you don't need to write these down, but you probably know these, and I just want people to kind of think about them. So you might think about ATP and ADP when you think of glycolysis. You probably think something about energy, which we've talked about how ATP is equivalent to biochemical energy because of the thermodynamics of breaking the phosphate bond, the phosphate diester bond. A lot of people think of pyruvate and glucose, which are the ending product and the beginning starting material of glycolysis. Sometimes I get people that think about NADH or NAD+. So that's one of the cofactors that gets used in one step of glycolysis. And that tends to be about the summary of everything. Some people know hexokinase, not just because I use it as an example a lot in the beginning of the semester, but because it's the first step of glycolysis, so a lot of people kind of think of that one. I've had some people think of the word mitochondria, and that's actually not where glycolysis happens. That's kind of a confusion where you've heard a lot of information very, very quickly. And so you tend to kind of cluster pieces of information together when they're covered together. And so actually glycolysis isn't happening in the mitochondria. That's the TCA cycle. So those are really probably where most people are as far as your brains and glycolysis goes. And so we need a lot more details than that. And I think we can do better. So we're going to learn about glycolysis the same way that we learned about the amino acids, in excruciating detail in a way that makes you not want to ever have to think about it ever again. So I'm going to take that and delete it, and we are going to start talking. Okay. So I will post these notes online for you so you have access to them, but I think that it would still be beneficial to you if you were to write them like I would be writing them on the board in class, because you just tend to learn more that way. All right, so if I wanted to do an overview, just kind of a very broad what happens in glycolysis, I'm going to give you a few pieces of information. Number one, the point of glycolysis is not to make energy. If it were, you'd be doing an incredibly poor job using glycolysis to make energy. It's a total of 10 chemical steps and you get just a couple ATP out of it. That's good enough for anaerobic or no oxygen cellular respiration, but again, that's not something that's going to be long term. You're just going to have to, kind of live with it and then die if you don't get any oxygen in a pretty short period of time. So I like to think of glycolysis more as the first 10 steps of breaking down glucose. And this destabilizes the molecule in a way that helps break it down into carbon dioxide. So we know that glucose becomes carbon dioxide in water, right? Like we've had to learn that reaction. That's probably one of the first reactions people memorize even before they really understand anything about chemistry. So we have glucose and we break it down into carbon dioxide. And that's about what I need you to kind of know for that. We use ATP gets used and made. So you'll have some steps where you have to put in ATP to make the reactions happen. But then when we're finished, we get a lot of ATP back. Well, not a lot, a little bit. We used NAD at one point to make NADH. This is useful for later on in respiration. So the TCA cycle, which is also known as the citric acid cycle, or the Krebs cycle. TCA stands for tricarboxylic acid cycle. I tend to call it the TCA cycle, but that's where you actually use that's not really accurate. So I'll just say that, but this is like later on in cellular respiration, like after you get a bunch of these cofactors during the TCA cycle, those are all going to go into the electron transport chain. We'll get there, we'll talk about it. Okay. I mentioned that there's 10 steps. There are 3 steps that are regulated. So I mentioned the other day. All right, bye Conrad. 3 steps that are regulated when you're talking about glycolysis. So out of the 10 steps, 3 of them are regulated allosterically. And so I think that that's kind of a good starting point. The way that I'm going to cover these steps for the next, well, for the next couple weeks, because it'll take probably 5 or 6 days for me to cover all these. As I'm going to talk about the terminology, like why do we name these enzymes what they're named, what the energy commitment is, why these were evolutionarily favored reactions, like how did these evolve to happen, the regulation, if they are regulated, the mechanism for the reaction, that kind of thing. So hopefully this will be okay online. I think that this information of anything else we've been doing is actually better online, or at least okay online compared to some of the stuff I'm having to do in my organic class. So that said, let's start with step 1. So I want you to think about, I'll go backwards. I want you to think about what happens when you eat sugar or any kind of polysaccharide that gets broken down into sugars. Okay, so if I eat glucose, it starts in your mouth, goes to the stomach, goes to the intestine, and when you eat these molecules, there are hormones in your body that pick up on the fact that you ate sugar. So one of those is insulin. And so the pancreas releases insulin. And insulin tells the cells in your body to take in glucose. Obviously, if you are a diabetic and you don't have insulin or enough insulin, this causes problems. Okay. But either way, your sugar starts in your mouth, gets into your intestine, gets absorbed by your intestine, floats around your bloodstream, your pancreas sees it, tells it to make insulin, your insulin tells your cells to suck in the sugar. Okay, so when we're in the cytosol, So the sugar that gets into the cytosol encounters the first of the 10 glycolytic reactions. All of glycolysis happens in the cytosol. So that's kind of an important thing to note because, like I mentioned, a lot of people think of the mitochondria, but that's actually later steps of cellular respiration. So in the first step, you basically have an enzyme called hexokinase. And it adds a phosphate to glucose. And glucose 6-phosphate is the product of this reaction. I tend to abbreviate that G6P, glucose 6-phosphate. If I need you to know, I need you to know what the words or the abbreviations stand for, but if I don't ask for them directly, you can always abbreviate them. So let's talk a little bit about the terminology here. Hexo. That is the prefix that stands for hexose, which is one of the words I talked about on Monday. That's a 6-carbon sugar, okay? So if you have a 6-carbon sugar, hexose, there are lots of hexoses, and hexokinase is not particularly specific about which hexoses it will attach to. So when we're talking about glycolysis, we tend to think about it connecting directly to glucose, but there are also forms of hexokinase that can attach to fructose and some of the other 6-carbon sugars. Okay? So the word kinase is kind of like, so the way that we name enzymes is to break them into classes based off what type of chemistry they do. So a kinase is an enzyme that adds phosphate using a molecule like ATP. And I mention like ATP because there are some similar molecules like GTP, UTP, CTP. They are all different phosphate carriers that we've evolved in different ways to use in different contexts. The vast majority of what you'll see is actually talking about ATP, but we'll see some examples, especially if you ever take Biochem 2 or if you take more advanced biochemistry in graduate school or medical school or something like that. So basically, all kinases are going to add a phosphate using something like ATP, some sort of high energy molecule like that. Talking about the evolutionary benefit, so back where I was talking about there we go, where you're talking about the cells taking in the glucose. When you have these transporters, and they basically will recognize glucose, and this can come in and out of the cell like that. Once you slap a phosphate onto the glucose, those transport proteins no longer see the glucose. And so it essentially traps the glucose inside the cell. So to make this happen, your body has to have released insulin. Your body wouldn't have released insulin unless there was sugar around. So it makes sense then that you have these kind of safeguards in place, right? You have to release the insulin to suck up the glucose. And at that point, you want to phosphorylate the glucose to keep it inside the cells and to keep it from leaving. So I'll just say adding a phosphate traps the glucose inside the cell, and it also starts to destabilize the structure of glucose for further breakdown. If you think about glucose as it is, there's all these carbon-carbon bonds. And you're essentially having to break every single one of those, right? And carbon-carbon bonds are not easy to break. You know that from organic chemistry. So this is one of those things where it's like you have to essentially, I don't want to say that. You have to think ahead of time about having to break all of these carbon-carbon bonds and how that's going to be facilitated. So when we're looking at the reasoning for some of these reactions, sometimes it can be very hard to tell why something would happen until you see a further step, 1 or 2 steps down. And so when those happen, I'll tell you, this doesn't make sense right now, but in 2 steps later, it's going to make more sense. Okay. So as far as the thermodynamics goes, this one has a delta G, and I don't have a delta symbol on here. So I'll just write a little d and a G. So the delta G, not prime for this, so at basically at biological equilibrium, it's about -4 kcals per mole. So this is very exergonic as far as biochemistry goes, okay? When you're talking outside of biochemistry, you could get things that are more exergonic or more endergonic, right? But in the sake of what we do in these aqueous solutions at ambient temperatures, this is essentially as good as it's going to get. You can't really get past -10, I don't think. So this is very exergonic. And so what this means is that the products will never build up in sufficient enough quantities to go backwards. In essence, this is an irreversible reaction. Okay, and so in biochemistry, if you have an irreversible reaction, that means you're basically going to have to regulate it, okay? So we can't just expect, right? I talked the other day about how we have all these branch points and things like that where things can go. We cannot just hodgepodge all molecules through every pathway because most of them are conflicting, right? Storing energy, breaking down energy, making sugars, making DNA, right? Breaking down DNA. All of these things happen in your body, and you have to be able to regulate which pathway things go through. So I talked about ways that we tend to regulate things. I mentioned reactions that use ATP tend to be regulated. This is a reaction that uses ATP. I said reactions that tend to have a very exergonic delta G, which also coincide with the ones that tend to use ATP. And then also the beginning point and branch points and end points of a biochemical pathway or a metabolic pathway tend to be regulated. So this one's going to be regulated. There we go. So this one gets feedback inhibition by the product, glucose 6-phosphate. And so one of the things that I want to do is stop and say, why does that make sense? Why would you stop at glucose 6-phosphate and say, when I get too much glucose 6-phosphate, I need to shut off this pathway? Well, there's like a giant truck outside right now. Maybe it's the garbage truck. Sorry, there's so much background noise, it kind of perturbs my brain when I'm trying to think. So if you're talking about glucose and going through and committing it to this pathway where it breaks down into making all this ATP, which is a long process, right? The first 10 steps of glycolysis isn't even halfway through the pathway. There's like another 15 steps that you got to go through. So when we're talking about committing it to this pathway, right? Glucose 6-phosphate, if you build up too much of that, your body's going to say, got plenty going through the pathway. We don't need this sugar in the cell. And so if you go back, as soon as you put that phosphate on there, which is irreversible, right? When you put the phosphate on there, it traps it in the cell. As long as you have too much glucose 6-phosphate, it'll say, we don't need any more of this in this cell, and that glucose can diffuse back out and go to another cell that needs it. So this kind of makes sense that you would be regulating it through glucose 6-phosphate. I don't know why I capitalized glucose 6-phosphate. It generally isn't capitalized, so just an FYI. It also gets regulated by genetic expression. Sorry, that I couldn't remember the word off the top of my head. So I want you to remember that pretty much everything gets regulated at the DNA level, the mRNA level. The allosteric regulation is exclusively what I'll be talking about when I talk about regulation. But remember that your body can regulate which DNA gets transcribed, which RNA gets translated into the protein product. Proteins can be broken down by the cell. They can be made by the cell as necessary. So you're also controlling things from that aspect. We're also controlling things through the hormones in your body, like I mentioned, insulin. So all of these things kind of work in concert together, but I want to be clear when I'm talking about regulation, I'm really only talking about this allosteric regulation of a small molecule binding to the protein and turning it on or off. I'm not going to talk about the kinetics of all of the reactions of glycolysis, but I am going to mention this one. There are multiple isozymes of hexokinase. And so that means that there's more than one type of hexokinase that exists in your body. And they have different affinities for KM for different types of 6-carbon sugars. So different hexoses are going to bind to the hexokinase in different ways. So I have a couple of examples here. You don't actually need to know the exact numbers, but this is just kind of how you would use this information, so it's a good example for this. The Km in most tissues for glucose is 0.5 millimolar, not milliliter, millimolar. In the liver, it is 10 millimolar. So, why do I keep putting milliliter? So, when we're talking about the Km, right, we're talking about the Michaelis-Menten kinetics curve, right? And I said that the lower the Km, the better the enzyme, the more tightly it binds to the substrate. So we kind of call this the binding affinity. At 0.5 millimolar in most tissues, you will reach 1/2 Vmax, right? So that's a lot faster and tighter binding than the one that is 10 millimolar in the liver. So basically, when you basically are having to feed all of your tissues, you're basically going to make sure all your tissues are fed and have enough glucose in them and have enough energy in them before your liver starts storing it as energy for further storage. So one of the things that happens with glucose after you've basically filled all your glucose up in your body, all your cells have enough glucose, you'll start storing it as glycogen in your liver and a little bit in your muscle tissue. And then also your liver's also where fatty acid biosynthesis happens. So if you have too much sugar and everything's fed, your liver's like, oh, we're now getting a ton of this glucose in here. We can store this as fat. Okay, so it kind of makes sense then that the liver would have a different version with a different Km. Okay, and then I want to do the reaction. And this is where I need to draw. Okay. So here we go. All right. So when I have glucose in the body, it is most often found in the cyclic form, not the linear form, even though it does constantly exist in equilibrium between the linear form and the cyclic forms, the alpha and beta cyclic forms. I kind of need a line there. I can't show the mechanism unless I draw that arrow, or draw that bond. And then I'm just going to draw my OH here, because I like to make sure that I remember that that has variable stereochemistry around that anomeric carbon. Yeah. Okay. So one of the things that is important to remember in your body is that you have plenty of acid and base floating around. You have water, which dissociates into H plus and OH minus. And you have plenty of amino acids that are going to be weak acids and bases. So it is very common in biochemistry to kind of think of generic B minus and H pluses when you're talking about amino acids doing certain types of chemistry. The way that I draw ATP is I'm going to draw ADP with a phosphate attached to it. And if you want to look up the structure of ATP, go for it. You don't need to know it for this class. I'm not going to make you ever draw it or memorize it or anything like that. And I'm not even having you in class in person anymore. Maybe I will have you draw it. I'll have you look it up and draw it on a quiz or something. One of the things that is going to be useful to this discussion is when I say we're making glucose 6-phosphate, it would help to know which carbons are which, right? So this is 2, 3, 4, 5, and then that little guy up there is carbon 6. So glucose 6-phosphate involves this OH up here in the top corner. Okay, so B minus, so some sort of generic base, which could be something like a deprotonated aspartic acid, so aspartate or glutamate inside the binding pocket. It could be something like lysine that's in its deprotonated state. It could be water, right? These are all possibilities when we're talking about B minus. Every protein has its own specific way, but I think using generic B's and H's and things like that is useful when we're just doing acid-base chemistry. So some sort of base in your binding pocket will grab that hydrogen, and then that bond will come up that. Try that again. There we go. Grab that phosphate, and then that phosphate will kick off ADP. Okay, so that's why I draw this as ADP with a phosphate on it. If I draw it as ADP with a phosphate on it, I can still show the active functional group and the chemistry that it's going to do. And so that makes it kind of useful. Oh my goodness, I put an extra bond on that phosphate. How embarrassing. Fix that. I'm glad I saw that. There you go. All right, and then I just have to add on my other OH groups. And my wiggle line and an OH. If you show it as the alpha glucose or the beta glucose. All of those are technically right. It doesn't really matter. I like to do it as the variable bond there. So that's the mechanism for the reaction. I guess I should have called that the reaction / mechanism. In these notes that I'll post online, I have the reaction at the top. Let's see if I can show you. So the very top of the discussion has that. And then I show the binding pocket in the mechanism there for the binding pocket. So you can see a little bit better of a version of that when you basically print off these notes or look at the notes that I'm posting online for you. Okay. And at that is the end of this discussion on the first step of glycolysis. At this point, you have glucose 6-phosphate. The molecule's trapped in the cell. The rest of glycolysis is good to go until we hit the next regulated step, which I mentioned there are 3 of those. So that is what I need you to know about glycolysis, first step. Stephen, are you still with me? That's kind of the end of what I had for this lecture. I usually do one step at a time. I usually do a lot more problem solving skill in this class as far as the mechanisms go, but obviously I can't walk around the classroom to see what you're doing, so that makes it a little challenging to do it that way. So I'm going to be presenting this material in just this format where I just give you the info and so just let me know if you have any questions on this. I guess we'll call that a day. Go back to my webcam. I'm going to be working with Brandon to try and figure out why my Wi-Fi connection isn't working very well. It might be like what Conrad said, where there's just so many people online right now, it's causing problems. But I don't think that would be an issue because plugging it directly in was fine. So I don't really know. And I should be the only one on my house Wi-Fi right now. So I don't know. We've had problems with the Wi-Fi in the past. I think I need a new router. But either way, this works for us. Oh, okay. That's it. All right. Well, I miss you guys. I miss your faces. I'll talk to you guys on Friday. If I can't get figured out what's going on with my Wi-Fi, I'll just be streaming in here. I know the lighting is terrible. It's all backlit, but it's this way to fit Brandon's drums and his harp and his rack of guitars behind me and everything else. So anyway, I will talk to you guys on Friday and email me if you have any questions. has been your biochemistry lecture for today.